So again, welcome and thank you for taking the time to join the webinar today, which will examine the coverage conundrum of multi-gene panel tests. I'd like to introduce my colleague and your speaker today, Dr. Jennifer Malinowski. Jennifer holds a PhD in human genetics and a master's in applied epidemiology and genomic concerns, both from Vanderbilt University. Prior to joining Hayes, Jennifer was research faculty at Yale University School of Medicine in the Department of Surgery. So at this point, I'd like to turn things over to Jennifer to dive into our topic today of multi-gene panel tests. Jennifer?
Thanks, Jim, for that wonderful introduction, and thank you all for joining us. So now that we have the housekeeping items out of the way, we'll get started. Today's objectives are listed on the screen here. We'll provide a little bit of a background for you on the technologies involved, covering multi-gene panel technologies. We'll move into germline mutations and tumor screening, and finally end with current recommendations and then the question and answer session. So examining many genes at once is easy and it can be very economical. But there are some things that you have to think about, like genetic disorders are really complex and hundreds of genes or dozens could be associated with any particular phenotype. There are thousands of multi-gene tests that are available, and the regulation is really limited. So CLIA certification and CAP accreditation are not the same as FDA clearance or approval, and most tests that we evaluate are lab-developed tests falling under CLIA. So the regulation is kind of like the Wild West. There's a few sheriffs, but it's a really large population to police. So this presentation will examine the key questions listed here about multi-gene panel tests. So what are the advantages and the limitations of these technologies? What's the difference between germline variant identification and tumor profiling? And how can multi-gene panel genetic tests be useful? So we'll start with an overview of the technologies that are used for these multi-gene panel tests. Sequencing technology began in the 1970s with Sanger sequencing. The development of polymerase chain reaction, or PCR, in 1985 really provided the technology that's necessary. The maturation of the next generation sequencing, or NGS field, really provided researchers with the ability to look at a variety of genomic alterations. So as you can see from this table, there are several technologies that can be used to examine the human genome. In the beginning of the genomic era, it was largely performed one gene or one variant at a time, and progress was pretty slow and really laborious. But with improvements in technology like NGS, researchers can interrogate multiple genes at the same time, significantly reducing both time and cost. And though Sanger sequencing has often been touted as the gold standard across sequencing technologies, you can see from this table that each of the technologies listed might be used depending on the desired result. So for example, Sanger sequencing can be used to detect variants from single nucleotide polymorphisms, or SNPs, to large rearrangements. But it really can't detect RNA transcript processing errors, certain regions of the genome, and it fails with allele dropout. It's also not really considered high throughput. And in fact, at a recent FDA workshop that was focused on next generation sequencing panels, experts in the field noted that alternate NGS platforms might be more ideally suited to verify another NGS platform than Sanger sequencing in some cases. So what's driving the explosive growth of next-generation sequencing tests? Well, as you can see from what we've highlighted, it's really the ability to assess large numbers of genes at once and the versatility of NGS methods that are partially driving this. So multi-gene panels are assays that interrogate multiple genes at the same time using next-generation sequencing technologies. So this could be gene expression assays, whole exome sequencing, or even a panel that looks just at 5 genes for a specific disorder. Despite the benefits of NGS multi-gene tests, there are some challenges based on the complexity of the technology that aren't necessarily present in older technologies like Sanger sequencing. And each step in the NGS process is an opportunity for error and should be considered when you order a multi-gene test. For example, DNA samples that come from blood are generally of higher quality than those that can be obtained from saliva or from buccal swabs. That can significantly affect your results. Errors or contamination from the sample preparation techniques, in the sequencing platform itself, in the data processing or analyzing steps, they can also contribute to the overall quality of the results. And each step builds upon the one previous to it. So if you have a problem, even if it's a small one and in the beginning of the process or a small one at each of the steps, your overall results can be dramatically affected. In general, there are three broad categories of limitations related to next-gen sequencing. They're methodological, analytical, and related to clinical utility. Most methods rely on PCR amplification, which is greatly affected by DNA material, as we mentioned in the previous slide, and pretty much are limited to short reads. In addition, the multiple platforms have varying degrees of sensitivity and specificity, and there's no real one best practice, even though there have been some suggested guidelines developed. There are numerous analytical limitations as well, including a significant computational burden that encompasses both storage and data processing. Bioinformatics pipelines differ not just across the NGS platforms, but even from one lab or one study to the next. So that makes comparing study results very difficult, since it might not really be an apples-to-apples comparison. And then finally, there are limitations related to clinical utility. The reliability of the test could be called into question if previous associations fail to replicate across laboratories. Secondary or incidental findings of deleterious variants in other regions of the genome might be reportable, but they require additional genetic counseling that patients may not be ready for. The clinical validity of every variant interrogated may also not really be established, which frustrates clinical interpretation. And most important is the impact of genetic testing on patient management. Will it change patient management? Will it inform at-risk family members? These are important considerations to look at when you're considering a next-gen sequencing multi-gene panel. So before I was lost to the audio here, I was asking, what are the challenges and limitations to next-gen sequencing technologies? Simply put, they are fast, high throughput, and less expensive per kilobyte of data than direct sequencing or genotyping could be for large projects. NGS has also matured since its emergence. The reliability improved substantially, and orthogonal methods like Sanger sequencing that have been used to validate NGS results are pretty much concordant. So for example, Myriad Genetics Laboratories' MyRisk panel is an NGS-based sequencing panel that looks at 25 genes associated with a variety of cancers. The analytical validity was assessed in a 2015 study where Myriad compared next-gen sequencing oncology panels. The FDA held a workshop at the end of February to obtain stakeholder input and expert advice for the development of regulatory standards for the tests in cancer patient management. The FDA notes that NGS tests are different from the typical in vitro diagnostic in that you can have hundreds, thousands, or even more variants to evaluate. And to evaluate each one could take years, and determining the clinical relevance could be very limited. So the FDA supports the use of representative variants to assess the analytical performance of an instrument and publicly available aggregated data to support clinical relevance for some tests. For example, the Illumina recently obtained FDA clearance for its next-gen sequencing MySeq machine. It's accompanying general assay and 2 cystic fibrosis tests using those methods. The FDA is actively considering regulation of lab-developed tests, moving from a position of enforcement discretion to active regulation based on the risk of the test. It's been estimated that more than 98% of next-gen sequencing oncology panels are lab-developed tests and follow CLIA guidelines. And so the test results from lab-developed tests are required to state that they're not from an FDA-approved or cleared test. This information isn't often known by the patient at the time of ordering, and differences between FDA and CLIA requirements may not be known to the ordering physician. Some professional organizations are making recommendations about the use of multi-gene panel tests for use in oncology for clinical practice. The American Society of Clinical Oncology recommends that providers with particular expertise in cancer risk assessment be involved in the ordering and interpreting of test results from these panels. The Green Park Collaborative, which is an initiative of the Center for Medical Technology Policy, further recommends that payers cover only those tests performed by labs with College of American Pathologists or CAP accreditation, and that panels of 5 to 50 genes, as long as 5 or more, are guideline directed and useful for the patient. But no consensus by the diverse stakeholders of the Green Park Collaborative was reached for panels containing more than 50 genes. So you might ask why the Hayes recommendation sometimes differs from those of professional organizations for genetic testing. And while we can evaluate some of the same scientific evidence, our reports follow a systematic framework and rigorous methodology to provide you with an unbiased recommendation. So between the FDA recommendations and those of these other organizations, it can be really challenging for payers to make sense of these multi-gene panel tests. We're staying at the forefront of these technological advances and changes in regulatory frameworks and responding to our clients' needs by evolving our genetics products, including our forthcoming genetics indication reports. You'll hear more about the indication reports and other changes to the GTE product line at the end of the presentation. We've mentioned CLIA a few times in previous slides, but we think it's important to highlight some relevant information. The Clinical Laboratory Improvement Amendments of 1988, or CLIA, established quality standards for all clinical laboratory testing to ensure the accuracy, reliability, and timeliness of patient test results regardless of where the test was performed. The Centers for Medicare and Medicaid Services, or CMS, regulates all clinical human lab testing through CLIA. And as we mentioned earlier, most genetic tests, particularly multi-gene panel tests, are lab-developed tests that aren't routinely regulated by the FDA and instead fall under the purview of CLIA. CLIA requirements are focused on the laboratory's quality control and methods. According to CMS, the CLIA Analytical Validity Review checks if the test finds what it is supposed to find. But this check can occur after the lab has begun using the test during its biennial review. The review also doesn't review clinical validation of the lab-developed test, the accuracy of the test to identify, measure, or predict the presence or absence of a clinical condition or predisposition in a patient. CLIA oversight is quite different from FDA oversight for tests. The FDA maintains regulatory responsibility for commercially available in vitro diagnostics tests. Tests that fall into this category are required to adhere to FDA requirements such as notification or registration, so 510(k) submission, or pre-market approval depending on their regulatory class. Clinical validity of a test is reviewed before the marketing of the test during the pre-market review. The FDA's Analytical Validity Review is more in-depth and comprehensive than the CLIA review, and it's focused on the safety and effectiveness of the test. The FDA pre-market review includes an assessment of the clinical validity. Others, like those submitted for pre-market approval or for some companion diagnostics, may need to provide additional evidence. New York is one of 2 states to be granted a CLIA waiver because their state laboratory licensing process is more rigorous than CLIA, requires tests that are performed on citizens living in New York to have clinical validity. So you may have noticed some laboratories have different tests to accommodate the New York license or do not test samples from New York patients. The College of American Pathologists, or CAP, accredits medical laboratories as well. This isn't required if the laboratory has a CLIA certificate of compliance, but accreditation by CAP also satisfies the requirements for CLIA certification, and many laboratories now carry both CLIA certification and CAP accreditation. The process for CAP accreditation is more intensive than what's required for CLIA certification, and it involves both the analytic wet bench process and the bioinformatics analysis process. CAP also handles proficiency testing for labs that require it. So it's important to understand that both CLIA and CAP are concerned with the methods being used and the quality controls of the laboratories. However, neither CAP accreditation or CLIA certification evaluate the clinical validity or clinical utility of the tests that they offer. Now that we've covered the basics about NGS technology, I'm going to transition into talking about germline mutation testing. There are different indications for germline testing. It can be performed as a screening test for carrier status, such as testing for BRCA1 variants in someone with a family history, but without a personal history of cancer. Germline testing can also be used to make a diagnosis for a specific disease. For example, a child who presents with intellectual disability having a genetic test to diagnose Fragile X. So though genetic testing for screening or diagnostic purposes can be used early in the setting of pre-implantation embryos, policy statements have been crafted by professional organizations about its use. So for example, there was a 2013 joint policy statement by the American Academy of Pediatrics and the American College of Medical Genetics and Genomics that didn't recommend routine carrier testing in minors if there's no clear health benefit in childhood. And predictive genetic testing for adult onset disorders should typically be deferred unless there's an intervention that, if begun in childhood, would improve overall morbidity or mortality. 5 professional organizations, including the American Congress of Obstetricians and Gynecologists, the National Society of Genetic Counselors, released a joint policy statement in 2015 on prenatal genetic testing, recommending that the condition being screened for should encompass at least one of the following. So a cognitive disability, the need for a surgical or medical intervention, an effect on the quality of life, a condition for which a prenatal diagnosis could lead to a change in perinatal outcomes or delivery management. The policy statement also acknowledged that in the case of multi-gene screening panels, or sometimes they might be called expanded carrier screening panels, healthcare providers could choose not to screen for some conditions and exclude certain other genes. For example, variants with high frequency and low penetrance of a phenotype, such as MTHFR, or for disorders with an adult onset phenotype, where the molecular testing is really unable to distinguish between a childhood or adult onset, such as alpha-1 antitrypsin deficiency. So the clinical validity of a test can be assessed by measuring its clinical sensitivity, specificity, positive predictive value, and negative predictive value. And these are easily calculated by using an epidemiological 2x2 table. So for example, to calculate the sensitivity of a genetic test, you'd identify the number of patients who have a disorder, or the truth, and then test positive for that particular variant. Divide that number by all the individuals with the disorder. If your test failed to identify a large number of patients in your cohort who actually had the specific variant, the sensitivity of your test would be considered to be low. There are several limitations to assessing the clinical validity for genetic tests. The most challenging of these is really the relative paucity of published evidence for clinical validity for any individual genetic test. So an example of this is Ambry Genetics' GYN Plus test, which is an NGS panel that assesses 9 genes associated with ovarian or uterine cancer, which include BRCA1, BRCA2, P10, and TP53. All 9 genes on this panel have medical management guidelines based on the identification of a deleterious variant. But a PubMed search done just last week failed to identify any articles that specifically mention the GYN+, which demonstrates how challenging it can be to perform a health technology assessment on these multi-gene panels. disorders with multifactorial genetic contributors, the role of environmental stimuli on disease, and variable expressivity and penetrance really contribute to the difficulty in accurately determining patients who have a disorder and those who don't, and those who could be at increased risk compared to those with normal or even decreased risk. Hayes' new GTE Indication reports will address the utility of genetic testing for a given disorder, taking into consideration all of these challenges. As we mentioned before, the clinical utility of a test considers how the test result will change clinical management of the patient, or in some situations like hereditary breast and ovarian cancer or Lynch syndrome, the medical management and surveillance of at-risk relatives. But what is the utility of genetic testing for a disorder with a late onset, or for a disorder where there's no cure or treatment? And the ethical, legal, and social implications of genetic testing also should be considered. And while cost effectiveness is not directly related to the scientific assessment of a genetic test, it might be a consideration for a specific patient. A recently published study looked at the cost effectiveness of an NGS panel containing genes that are associated with Lynch syndrome and other highly penetrant colorectal cancer syndromes. The authors found the use of the panel increased survival by nearly 2 months, 0.128 quality-adjusted life years, and $4,650 per patient. This resulted in an incremental cost-effectiveness ratio of $36,500 per QALY, compared with standard care, and a 99% probability the panel was cost-effective at a threshold of $100,000 per QALY. So let's compare 2 tests for the same indication, carrier screening for cystic fibrosis. Cystic fibrosis is an autosomal recessively inherited condition that affects about 70,000 patients worldwide. It's estimated, though, that millions of individuals are carriers of the mutated CFTR gene, which is responsible for the disease. And because carriers don't exhibit any symptoms of disease, many of them are unaware of their risk for passing cystic fibrosis to their children. There are many tests commercially available for cystic fibrosis carrier screening. The overwhelming majority of these are lab-developed tests, which are regulated through CLIA. As you can see by this comparison of 2 tests, Progenity's CFNext and Illumina's MySeq DX cystic fibrosis assay, there can be very stark differences between 2 assays for the same indication. And though it may be tempting to automatically choose an assay that tests for the greatest number of variants for a particular disorder, the additional information may not actually be clinically meaningful, and it may not even be clinically validated. So as we wrap up this section, just take a moment to review the main issues associated with NGS multigene panels. There are multiple technologies with varying capabilities to identify variants. So to understand which is the most appropriate test for your patient requires knowledge of what the limits of the test are, the analytical validity of the test, the clinical validity of the test, and importantly, how the test results are going to change patient management. We noted significant limitations of genetic testing with NGS panels, including identification of variants with unknown clinical importance and potential uncertainty surrounding the best choice of a test for a given patient. Other challenges to evaluating the clinical utility of a test include the impact of genetic heterogeneity and the environment on disease expression. Variable expressivity and incomplete penetrance of a disease, even within a family, can result in misclassification of individuals at risk or affected. Genetic testing for late onset disorders, or disorders for which there is no current treatment, and the ethical, legal, social issues of testing are further complications. And it's really from this standpoint that we've developed our new GTE Indication report, which is an overview focused on evidence for clinical utility of genetic testing for a specific indication, such as hereditary breast and ovarian cancer or autism spectrum disorder. And we'll discuss this in more detail at the end of our presentation. So now we'll switch gears and focus on tumor profiling. So many of the same issues that we identified with germline testing, like the difference between FDA-regulated tests and those monitored by CLIA as lab-developed tests, are equally important in tumor profiling tests. Before we discuss how tumor profiling is currently used, we should probably explain what we mean by that. So while germline variants are inherited, tumors often accrue somatic changes to their genome. These changes can include point mutations, duplications, insertions, copy number changes, even structural changes like breakage and fusion of chromosomes, creating fusion genes or proteins. NGS, from custom panels to complementary DNA microarrays to whole genome sequencing, are capable of identifying these types of biomarkers. Tumor profiling is used to help providers convey the prognosis for a particular patient. So, for example, through tumor profiling, multiple subtypes of breast cancer have been identified, including luminal A, luminal B, and HER2 enriched. Luminal A subtype, which accounts for a substantial fraction of all breast cancers, has a generally favorable prognosis, while luminal B tumors are associated with increased risk for relapse and a poorer prognosis. Tumor profiling can be used to predict risk of recurrence or metastases as well, based on the underlying biology of the cancer. And it might complement more traditional prognostic clinical pathologic features in order to provide patients with a more accurate disease trajectory. Tumor profiling can inform treatment decisions for patients as well. So using our example of breast cancer, patients with HER2 overexpression, or simply HER2 positive, can be treated with drugs that specifically target that, like Herceptin or Tykerb. But patients who are HER2 negative are really not likely to respond to those drugs, and they might require chemotherapy or an alternate treatment. So tumor profiling is really being emphasized now in basket clinical trials that categorize patients by the molecular profile of their tumor instead of by the type of cancer that they have. So the National Cancer Institute's Molecular Analysis for Therapy Choice, or the NCI MATCH program, is an example of this new kind of design for a clinical trial. So they enroll patients with a variety of cancer types, so breast, colon, lung, or prostate, and use tumor profiling to group them for potential treatments based on the tumor profile. So in one trial, you could have someone with breast cancer and someone with lung cancer, but whose molecular profile of their tumor is similar, and they would be tested on the same treatment. In August 2014, Hayes published a comparison of available tests that provided predictive information regarding the recurrence of primary breast cancer. So as you can see from the excerpt from this report, across 5 tests for breast cancer, there are 4 technologies that are used, and there are significant differences in the number of biomarkers assessed, from 5 proteins by mass spec to 7 genes by MammaPrint. 2 of the tests are FDA cleared, while the other 3 are lab developed tests. And while not included on this chart, varying endpoints and the fact that most variants tested on these assays are specific for each test, meaning there was very little overlap between the tests. And that has really important implications for the clinical validity of each test and makes doing an apples-to-apples comparison of these tests inappropriate. New lab-developed tests coming on the market, it can be really challenging for providers to select the best test for their patient and for payers to develop an evidence-based coverage policy in a timely manner. So Hayes offers multiple solutions for our clients, from our client-requested laboratory comparison report to our new GTE Synopsis to more in-depth GTE reports, indication reports, to help you navigate this process. Similar to the challenges faced in comparing methods for germline testing, tumor profiling tests can be performed using multiple methods depending on the type of variant you're looking for, and the number of biomarkers or genes evaluated can vary substantially from test to test, just like we saw on the previous slide, even when it's the same clinical indication. So as you've seen from this presentation, genetic testing and tumor profiling are very complex and emerging technologies that are pushing boundaries pretty much every day. You might have seen a report that was published in January by Forbes that described a new collaboration between biotech company Illumina, Bill Gates, Jeff Bezos, and several other venture capital groups called GRAIL, where their goal is to create a single blood test able to detect any cancer type, so a pan-cancer test. So when and if this project succeeds, it's going to be really critical to put the clinical validity and utility of the test into context. And this is an example of a solution that we would be able to help you with. You may have also seen this other widely publicized news article that took place in January that described Independence Blue Cross's coverage of whole genome sequencing and a diagnostic proteomics panel, GPS Cancer, from Nat Health for patients with cancer. The GPS Cancer panel is designed to identify variants that are driving the cancer and predict the best choice of treatment for the patient. But despite the assertion that this is really moving, shifting whole genome sequencing from research to the clinical setting, the collaboration is actually driving investigational phase 2 trials. And as seen from previous slides, we've described there's a lot of current methodological, analytical, and clinical challenges that are being faced in implementing clinical genetic testing and tumor profiling. So the utility of this is going to be quite interesting to observe. Hayes' evidence-based reports really sift through the hype, though, to identify the analytical and clinical validity and clinical utility of genetic testing, which gives you the evidence you need to make the informed decisions for coverage policies and for patients. And I just wanted to highlight here the explosion in literature that we have seen in the last several years. And we took just one indication and one type of test, so whole exome sequencing for neurological conditions in a pediatric population. So fairly stringent search criteria, but as you can see from 2010 with only 2 articles to 2015 with almost 200, this dramatic increase in literature makes it really challenging to go through and identify, again, the clinical and analytical validity and the clinical utility, trying to help you figure out when a test might be appropriate for a patient. So with that, I'd like to share Hayes' recommendations regarding multi-gene panel tests. As we've highlighted, they can be used for many different types of indications. They can be conducted on different platforms, have different bioinformatics analyses and output, and there's very little regulation around the lab-developed tests. In short, it can be very challenging to ensure that multi-gene panel test being ordered by a physician is valid and can improve patient management. So to this end, we'd like to recommend the following guidance when assessing panel tests. First, the assay process, which is from how samples are collected to how variants are called and confirmed, is an important aspect of the panel test. We'd like to see an established assay process to help ensure that the results are analytically valid, and laboratories need to be transparent with all aspects of their process. Second, each disease and population needs to be assessed to determine when and for whom multi-gene panel testing is appropriate. And it ultimately should be shown that the results of these tests will change patient management. Finally, other factors such as efficiency and cost should be considered. For example, multi-gene panel test reimbursement levels should be set to no greater than the combined reimbursement level of those specific gene variants that have documented utility. So we hope that these general recommendations can help guide your assessment of multi-gene panel tests. And it's important to note that multi-gene panel testing is just one example of how the genetic testing field is rapidly evolving. And to this extent, Hayes is also enhancing our product line to meet these changing needs. At this point, I would like to turn it back over to Jim to discuss those for you.
Great, thanks, Jennifer. This is a complex topic, and I think you shed some light on the issue, so thanks so much. When we started the GTE program back in 2007 here at Hayes with GTE Synopsis, reports, and lab comparison tables, we really couldn't have anticipated the amount of growth that occurred, as Jennifer pointed out earlier, and especially in the last few years. So we knew that a change was going to be necessary in our GTE program. So what we do is we work with our clients to develop the next evolution of the GTE program here at Hayes. In this past December of 2015, we launched a new synopsis format, which now includes a brief and an at-a-glance section, as you can see in the photo here on your computer. Also, information such as costs, CPT codes, and methodology are now available in an easy-to-view table format. However, we knew that this enhancement was not going to address a root question that we keep hearing over and over again, which is, will it change patient management? To answer that question, we needed to develop a solution that was based first on the clinical indication as opposed to specific genetic tests. So our latest innovation, GTE Indication reports, has been developed to do just that. These reports will look at clinical utility for a specific indication. And with clinical utility established, specific tests can then be assessed. This unique solution addresses not only a main concern of our clients, but also allows for flexibility for each client and their unique needs. We will also be able to keep on top of the most up-to-date literature around the utility of genetic testing while allowing our clients to continue requesting specific tests of interest. And our intention is to launch this, the GTE Indication report this summer. So at this time, we would like to open the floor for questions. And just as a reminder, you can type your questions into that question box as shown on the screen here, and just hit that send button. If we don't get to your question, we'll reach out to you via email with a response. So the first question I see here is, Jennifer, if you can answer, if we request a synopsis for a test and the Hayes rating is D2, but we hear from physicians that there may be utility for a genetic test, can we request an indication report? If so, how will they be prioritized?
That's a great question. And so what we do on the GTE team is regularly evaluate which reports are requested to identify patterns. So if we see that a report for a specific test is being requested multiple, a lot, or even for a particular indication. So if we see a lot of interest around, say, autism spectrum disorders or another indication, we use that to help us drive prioritization of our reports. And so even though an individual test might have been a D2, if you see or if you feel that your providers are indicating that there is clinical utility for genetic testing for a particular disorder, please let us know. Send us a request, send us an email, and we can use that to help us prioritize the indication reports then.
Thanks, Jennifer. One question I'd like to address, I know we get a lot of questions about the recording and the slide deck, so all of that will be available after the conclusion of the webinar today. So if you're registered for the event, you'll be receiving the recording and the slide deck. Another question that came in is, what is the proper coding for NGS panels that do not have a specific CPT code? Some examples, 81479, 81407. It seems that stacked billing of individual Tier 1 and Tier 2 CPT codes when only one test is performed is not appropriate. But that is what we are seeing.
That's a great question, but I unfortunately don't have an answer for that right now. We'd be happy to look into that and help you identify ways to deal with that that Hayes can help you with, but unfortunately I just don't have an answer for that right now.
Okay, no problem. We'll have somebody follow up after the webinar to the person with that question. Jennifer, how about, early in the presentation there was a chart with different technologies listed and what they can do. Do you have a paper or something on this? And do you include this in the report so we know when a test isn't the right one for the indication?
Oh, that's an excellent question. So to the first point, we are actually planning a webinar later this summer and an accompanying white paper to really describe in detail next-gen sequencing technologies and when each could be used. So really looking at, what kind of genetic alterations they are able to find and are good at. As far as whether or not it would be included in reports, well, for synopses and health technology assessments, it probably would not be. Those really are looking at just an individual test, and so it wouldn't be appropriate to really give an overview of the different kinds of technologies, since we're only looking at one. But it might be included in an indication report, and I think that would really be very topic dependent. For an indication with a wide variety of genomic alterations or many tests that look at different types of things, then that would probably be an appropriate place to have a table like that and to discuss the limitations and benefits of each of the technologies. And that would be something that we could put into a report as necessary or if client requested.
Thanks, Jennifer. Another question coming in. The question is, do you also cover for pharmacogenetic testing products?
That's a great question. I believe we have maybe a few reports on the Knowledge Center right now, but if it's something that is of particular interest to your company, please let us know if there's an individual test that you're getting a lot of interest in, or if you're looking at for a particular indication, then that might be something that an indication report would be appropriate for. So it would be really great if we hear that from you. We can certainly work to prioritize. So please send us an email and we can work on that.
Great. Questions are rolling in now, Jennifer. So
great.
Next one is, you talked about the differences between FDA regulation and CLIA, C-L-I-A. Are you saying the tests that are from CLIA certified labs aren't valid?
Oh, that's a really excellent question. I'm glad somebody asked about that. No, that's actually not what we're saying at all. CLIA certification and FDA approval or FDA clearance are very different mechanisms for tests. And so they have different requirements, different levels of evidence that are required to be included. And so it's really important to understand what each of those brings to the table. Just because something is CLIA certified or a lab developed test does not at all mean that it isn't clinically valid or analytically valid. And this really sort of emphasizes, though, the need to understand the differences and to look at each test individually. When we perform a health technology assessment, we look at the analytical validity, the clinical validity, and the clinical utility. So if a lab-developed test has all of that information available for us to be able to assess, then it very well could receive a very high rating simply because we're able to evaluate it then. And so it's really, we're looking at the published evidence and what there is. So we're not at all saying that one is better than the other. We're looking at a body of evidence.
Great. Next question. How will you weigh the importance of changes in surveillance surveillance in regard to clinical utility versus changes in direct management in new reports?
That is probably something that would have to be covered on a very topic-dependent level. So for some indications, so for example, hereditary breast and ovarian cancer, where you could identify in one, in a family that there's a specific variant, and then you could then look at individuals who are asymptomatic but who carry that variant and follow them, and they would be eligible for increased surveillance and perhaps prophylactic measures. But that's something that really you'd have to look at individual indications, and it would be covered in reports as appropriate.
Okay. Another question. When there is, quote unquote, enough published evidence to conduct a GTE, unquote, what does that mean to the rating of the test as opposed to a D2 rating? Still considered experimental, investigational until more evidence for standard of care?
So for a D2 rating, the evidence just isn't published and we can't make a determination. When we are able to give a test a different rating, those ratings correspond to the type of evidence that we've been able to assess. Beyond that, I'm not sure that we can say that there's a certain threshold that has to be met, like X number of studies that have to be published to support a specific test. It's really looking at the body of the scientific literature and assessing the assessing the quality of the studies that have been done and the evidence that they publish.
Okay. Next question. How would you rate a 7-gene panel test in which 3 of the gene tests are clinically useful and valid and 4 are not?
Oh, that's a great question. So from our perspective, as we said a few slides towards the end, our recommendation would be that reimbursement would be set no higher than the limit that you would have for each of those individual tests. So for example, in that panel, if 3 of the 7 genes are clinically useful and appropriate for that patient, for that indication, then reimbursement for that should be set no higher than individual testing of those 3 genes. One of the challenges with next generation sequencing is that oftentimes you can have a panel, excuse me, you can have a panel where just because of scale of economy, it may be less expensive to to test using a panel than to test using each individual gene sequentially. But that's something that is very test specific. So if you are seeing this in your work and it's for a test that we don't currently have a report on, we're happy to take client requests for synopses and evaluate more specifically for individual situations.
Okay. A couple more questions I think that came in. So the next one, will the indication reports also address the various types of methodologies that can be used to perform this test? And which ones would be considered higher quality tests for that particular evaluation?
Another great question. Excuse me. So for certain indications where it's appropriate, we will provide a table of the various technologies that are used, as well as what their limitations and their strengths are when they might be used. You could easily imagine that a test could look at germline mutation, point mutations, and then you could have another test for the same overall indication, but it's looking at gene expression levels. And so depending on what you're looking for would determine, what technology you're going to use. However, we're not going to be evaluating specific tests in our indication report. So it's really more of an overview of the technology and types of technologies that might be used, but not getting into individual tests. That's really where our synopses and our reports are for, which are client-driven. We also have the ability to do client requests for lab comparisons or for test comparisons. So if you're really looking to try to get at which one is most appropriate for whatever indication, that's something that we are happy to take on. Please contact us and let us know what your needs are, and we would be happy to address that.
Great. Next question, Jennifer. Some determinations are held as national coverage positions and some aren't. There's always a bit of confusion there, so I was wondering if you plan to address that in the reports.
All right. Well, we really do only look at payer coverage policy on the national level. We can't really get into the regional decisions and the reports that we write simply because of the number of plans that we provide services for. Again, if that's something that you have a specific need for, then we'd be happy to, work on that with you. So please send us an email and let us know what your needs are. That, if that's a concern of yours, please let us know.
Okay. Another question: does Hayes look at CMS, NCD, and LCD determinations for a test?
Right. So like I just mentioned, we look at the CMS, the NCD, so the national coverage decisions. We don't look at the local ones.
All right. That's almost near the top of our hour. So there's a few more questions in the hopper there that we'll reply to. It's just to give everybody a few minutes to probably get off to their next meeting. Thanks a lot for joining us, and on behalf of myself, Jennifer, and everybody here at Hayes, I'd like to thank you for participating in the webinar.